Okay guys, welcome back. We are on the homeward stretch, believe it or not, even though it's the end of the third week. We are looking at just 3 more objectives. Maybe it's more like 4 more because we're going to have to backtrack for a couple objectives I skipped. So let's say 5 more objectives that are left. We're going to look at the organelles, the compartments that we left off with last time that are within the cell, okay? Again, this is all chapter 3 kind of stuff, all right? So this would all relate to Smart Book Assignment 3 that we have up on Connect. So let's move down through the organelles. We're going to need to know the basic functions and structure of these various organelles, all right? Again, you want to think about this cell almost like it's your own body, right? It's going to have a boundary with the plasma membrane, just like we have a boundary with our skin. It's going to have various organelles that perform specific functions, just like the organs within our body. One difference would be that in a cell, you can have more or less of certain of these organelles depending on the actual role that that cell is playing in our body, okay? So, first thing off, everybody knows about the nucleus, all right? So the nucleus is basically the control center of the cell, right? It's the easiest one to see, and actually, it's actually easier to see when we stain a cell, especially when we're looking at a light microscope. And a lot of the stains we came about that we used to stain the cell actually came from the textile industry. So when textiles really took off, when we started seeing all these different colors and dyes that were used, some geek in a lab, like me probably, said, well, we can't see anything with these cells. When we look at our preparations, let's try staining them. And lo and behold, all these things lit up. The nucleus most of all. Okay, so the control center of the cell is the nucleus. And within the nucleus we have that organic compound, nucleic acids. And in this case we're talking about the DNA. Okay, we'll talk later more about the DNA and why it's called D, it's for deoxyribo. But we'll visit that again later. Right now we just want basic functions. And what do we find in this organelle? Well, we find DNA, which is basically a genetic library, okay? With very few exceptions, every cell has DNA, and those cells have the same DNA, all right? Unless there's been some kind of mutagenic occurrence or where there's a mutation of the code. And remember, the DNA is made out of nucleotides, right? So the order of these nucleotides matters. And unless there's some mutation like that, all of the DNA and all of the cells that we have are identical, okay? Now, the important thing about being the control center is that the DNA in this nucleus actually dictates the kinds and amounts of proteins that are going to be produced. So here we see this connection. There's a connection between DNA, this nucleic acid, and proteins. We'll revisit that in the beginning of Module 2. But for right now, we're just getting the bare bones down. Okay? We see that there is a double membrane associated with the nuclear envelope. So this plasma membrane we've been talking about, you can see here where I'm kind of drawing here on our nucleus, that there's actually a double membrane. And there are pores in that double membrane that will allow items to move through, okay? But this is a double membrane, so this is kind of double what you would see on the plasma membrane that surrounds the cell, okay? All right, so here you can see that there are actually pores in that double membrane, and we'll talk about what those pores are for later, okay? We also have, as another organelle, ribosomes. Now, ribosomes are going to play a role of protein synthesis for us, okay? So we're going to find that there's a connection between the DNA that codes for proteins and the location where those proteins are produced. Okay, so we have the code in the control center, the DNA being the code, and then we have the actual production of proteins that occurs at ribosomes. Okay, so you want to remember ribosomes. When you're studying, you want to put off to the side really short, descriptive definitions you can remember, right? So for the nucleus, it's the control center with DNA, right? Codes for protein. And then for ribosomes, I always think protein factory. Okay, it's taking that code, making proteins out of it. And we'll find that ribosomes are actually found throughout the cell. They're found free. That means that they are actually kind of, not really, but they're almost like they're free floating within the cytosol, within the liquids. Okay, and then we have the membrane-bound ribosomes. And these membrane-bound ribosomes are attached to membranes that are part of the next organelle we're going to talk about. Okay, so ribosomes, protein factory, reading that code that we get from the DNA. All right, so where are many of those ribosomes? Many of those ribosomes are actually attached to something we call the rough endoplasmic reticulum. Okay, now this is a labyrinth of membranes that exist outside of the nucleus. Okay, and these membranes are studded with ribosomes. They're just kind of scattered with ribosomes all around it. Okay, and those ribosomes, of course, are making proteins. Okay, so these proteins are going to exist and function in a certain place if they're made at the rough endoplasmic reticulum. Some of them are going to be shipped out of the cell, so we'd say they're secreted. Okay, some of those proteins are going to exist on the cell membrane, so let's think about those. You might run into the channels, right? The pumps we talked about, the cell identity markers, or cell adhesion proteins like for tight junctions. Okay, and some of them might go to what are called lysosomes. Okay, now the lysosomes then are yet another organelle that we need to talk about. Okay, so rough endoplasmic reticulum, what makes it rough? It's studded with ribosomes. These ribosomes are attached to that membrane. Okay, and this is where we're making certain of the proteins that the cell needs to make. Okay, some of them are shipped out, secreted. Some of them are stuck to the plasma membrane ultimately, and some of them are going to go to this lysosome that we need to talk about. All right, now any of the ribosomes that are stuck to the rough ER are ultimately going to have to be shipped appropriately to the different locations within the cell or outside the cell. Okay, and that's what the Golgi apparatus is all about. Okay, the Golgi apparatus is like a FedEx, or if you prefer UPS, right, or whatever, right, or USPS. So this is basically a labyrinth of membranes, another set of membranes that are actually going to take any proteins that come from the rough ER, and they're going to direct those proteins in the places they need to go. They're going to ship, tag, stamp those proteins for where they need to go, right? So just like you have a barcode on something that comes from FedEx and it makes sure that it gets on the right truck and goes to the right house, this is what the Golgi apparatus is about. Okay, so we have the rough ER with ribosomes making proteins that go to certain places, and then we have this Golgi apparatus, which is like a FedEx that's going to take that product, that protein product, and ship it where it needs to go. Okay, so here we can see what those actually look like. Way back in the 50s, if you can think about that, the 1950s, we saw these things in electron microscope images, and we had no idea what they were doing. But now we do, okay? All right, so one of the locations that proteins that may be shipped through the Golgi apparatus will go would be the lysosome. Okay? Now, lysosomes are actually like the recycle center of the cell. Okay? Lysosomes are going to be containing digestive enzymes, so proteins that happen to be enzymes that are produced may be shipped to the lysosome, and the lysosomes are going to be a place where we break things down. Okay? Now, what kinds of things are we breaking down, you might ask? And so to answer that question, I would mention to you certain cells in the body called phagocytes. Okay, so we have phagocytes, meaning eating. So think esophagus. The esophagus, the phago part of esophagus, is about eating. Okay, and these are cells that eat. Okay. So when we look at certain cells that gobble things up, like cells in our immune system, let's say they're gobbling up bacteria, or a dead cell, okay, or something like that. These cells are going to have many, many lysosomes that will help them to digest those various things, okay? So digestion could proceed safely in these compartments of the cell without devouring other sections of the cell, right? These enzymes are very active. And so what are they going to digest? Well, in this case, bacteria, debris, dead cells. But it's also true that digestive enzymes within lysosomes also will break down maybe the cell itself. If a cell needs to dispose of itself because there's something wrong with the cell, okay, that also does happen. Whole organelles can be actually devoured by the lysosomes. All right, so other organelles besides lysosomes can be devoured by these enzymes, all right? And there's a lot of different functions that they can play. All right. Another set of organelles that really are not a membrane-bound organelle. Everything we've talked about up to this point, with the exception of the ribosome, are membrane-bound organelles. We've got the nucleus, two membranes, in fact, around that. We've got the rough ER, that's a membranous organelle with ribosomes attached. We've got the Golgi apparatus, membranes as well, and the lysosome. Well, here we're talking about something that is more of an infrastructure, a skeleton in a way, for the cell, okay? And so for good reason, we have a term that's very descriptive, cytoskeleton, right? So it's a skeleton for the cell, okay? A lot of people will think about a cell, and pictures even that your textbooks have, that would suggest that the cell is like just a sack of liquid, right? It's cytosols in there. It's mostly a liquid contained compartment there. But actually, a lot of these organelles we're talking about are positioned in various places within the cell, and they don't just float around randomly, okay? Some do, but very many of them are held in place by a cytoskeleton. So what I like to think what I like to have people think about is about your house, right where you are at home, or a building, right? You're going to have an outside surface to your house, okay, and walls that will give structure to those outside surfaces. But then on the inside, you also have walls, and you have rafters in your roof, and all of that not only may give structure to the outside surface, like the peak of your roof at your home, but also give structure to the floors in your house. If you have a couple, 3 floors at your house, they need structure, okay? And when you think about it, moving around your house also requires hallways, right? Hallways that take you to specific rooms in your house. So a cytoskeleton gives you many of these very same things when it comes to a cell, okay? So we've got 3 different cytoskeletal structures I want you to know. Microfilaments, okay, would be the first one. Here we see some microfilaments that are in green here for this cell, and these are a very special kind of microscope that would give us these images. A confocal microscope would give you this kind of fluorescent look to the cells. And what they do is they use molecules that have a fluorescent molecule on them, okay, that would bind to the, in this case, actin subunits, okay. Actin is a protein that makes up microfilaments, okay, and microfilaments are all about cell shape, all right. So if you want to, again, attach short, simple definitions, microfilaments give a cell shape, all right. Now, let's think about a second cytoskeletal element called the intermediate filaments. Okay? Well, intermediate filaments are more about their fibrous nature, which gives them strength. Think about fibers that would be in a rope. Okay? Now, if we look at the actual picture here, it looks like a rope, doesn't it, kind of? All these different strands, right? We have 1, 2, 3, 4, 5, 6, 7 strands in this particular intermediate filament, and it looks like it's kind of bound up and twisted like a rope, and ropes give you strength. So we can talk about microfilaments with giving you cell shape. We can talk about intermediate filaments as giving strength. Now here you can see that there are intermediate filaments all over in this particular image in red. Okay, so they've used a red molecule here that will bind to the fibrous subunits of an intermediate filament, and it shows you that intermediate filaments give strength throughout this cell. Okay. Finally, we run into something called microtubules. Okay, now microtubules here have actually been stained in green in this particular image, and microtubules play a couple different roles, but the one in particular that I'll mention here that you might want to remember would be that these actually act like a superhighway within the cell. Okay, so these are the hallways within the cell. These are providing a mechanism for movement in the cell by which we can move specific items around as we would need to, right? A cell is doing that, believe it or not. It's not just a sac, just a blob of liquid there, right? It's got shape, it's got strength throughout it, and these together, these cytoskeletal elements together hold organelles in place. But then we also have the ability to move things around within the cell. So I think about the hallways in your home, right? And I think about this superhighway that happens to be in the cell. It's kind of the same basic idea. Okay, so these are acting like the bones and muscles of your cells. This is a picture of one of those images with a cytoskeleton being stained in yellow. I did a bunch of these back as a grad student at Binghamton, at BU, and I did a few, and they were pretty cool. I didn't do a lot of it, but those instruments are mighty expensive, and they give you some pretty cool pictures though. Okay. All right, we're going to hit one more organelle that I would like you to know about before we get into the mitochondrion. Okay, the mitochondrion we're going to spend a bit more time with, but this is our last one for now. So we had a rough endoplasmic reticulum, and now we're going to run into a smooth endoplasmic reticulum. All right, so rough endoplasmic reticulums, they're going to be rough because of ribosomes that are attached to them, and those ribosomes have to do with protein production. Okay. Smooth endoplasmic reticulum, well, this would be a labyrinth of membranes that actually have nothing to do with protein synthesis. They're not rough, so they don't have those ribosomes attached, right? But this kind of labyrinth of tubules or membranes will play a role in many other functions that have to do with lipids and detoxification. Okay? So when you think of an organ, let's just take ourselves out of these little cells and think about what organs are involved with detoxification. One of them you might think about would be the liver. Okay? It so happens that liver cells are chock full of smooth endoplasmic reticula. Okay? They are all about detoxification, and one of the things you might think about is alcohol, right? Alcohol is going to be broken down, and these compartments of cells have a lot to do with that. But they also have a lot to do with metabolizing lipids. Okay? So when we say metabolizing, we mean that's breaking things down and building things up when it comes to reactions in a cell, right? Well, this particular subcellular organelle has to do with making certain kinds of lipids and with breaking down fats as well. So these cells would be involved with breaking down fats if you have too much fat in your diet, for example. These particular organelles would also have something to do with the synthesis of cholesterol and steroid hormones. And to make those, well, you need raw materials from lipids. Don't forget, a while back we talked about how cholesterol and steroid hormones are actually just a few steps away from fatty acids, okay? So this is important for the production of hormones that are actually essential for normal function. So I don't want you to think about fats and the metabolism of fats as a bad thing always. We need fats. We need these steroid hormones. Cholesterol is a normal component of our plasma membranes. So these are all good things to have, all right? Now, one thing on the kind of like the negative side of this, right, is when we think about fats and growing a tolerance to something, like if it were alcohol or if it were the intake of fats, well, it so happens that cells of the liver are very capable of growing more smooth endoplasmic reticulum within their cells. Okay? So as you imbibe more alcohol, perhaps, or as you take in more fats, what you do is you trigger your body to respond to those demands, and your liver will actually, grow larger cells with more smooth endoplasmic reticulum within them. Okay? And this is a big reason why tolerance exists. You can tolerate more alcohol because you actually have bigger liver cells chock full of these smooth endoplasmic reticulum. And actually, if you look at someone's liver that's been taking in too much fat or taking in too much alcohol, their liver will be much bigger because it's just responding to the needs and the demands placed on it. All right, so I think this is probably a good place to leave off. We've talked about a lot of the organelles. Maybe before we do the mitochondria next time, I'll do a quick review of these various organelles again, okay? But I think this is a good place to leave off. I think we've attacked at least 3 of our last objectives, and then we're going to go back and address a couple we've skipped, okay?